not have heard about PFAS, but if you go to your phone in the Google News and search PFAS, a lot of news will pop up. Some of them about the political influx that we have regarding the PFAS regulation, many of them about human health, and some of them about environmental health, how it will impact the microbial community or in general human health or ecosystem. So, but what are really PFAS? PFAS are polymer or chain of carbon that's shielded with fluorine. Carbon and fluorine creates strong bonds that nothing can break this bond. Not even microbial community, not even extreme heat. So it means very resistant. And that's why we call it forever chemicals, because they are not readily biodegradable. And they make a problem in the environment. Everything around us has been formulated with this chemical, with PFAS, because it's very resistant and make your fabrics or any material, coating material in front of you, make it hydrophobic and make it resistant to the heat. And you can see that their half-life in the environment is really high, between 8 years to 111 years. That's why we call it forever chemicals, because they are not really readily biodegradable. And you can see the picture of polar bear here. Scientists, they use it as an environmental example because they claim it has been also found in blood of polar bear as extent of contamination. So, but when we say that it's very strong and it's resilient to the heat, one main application comes in the picture. What is that? Firefighting foams, right? US has widespread military bases in the whole country or even other countries in the Middle East. So all military bases, they go through regular firefighting training. And every week they release high quantity of PFAS to the atmosphere. Some of them, they go directly to surface water, but some of them to the soil. But eventually all of them, they find their way to agricultural soils that contaminate our soil, that's the main media for producing the plants. Many things can impact their fate in the vadose zone or in the soil, including microbial community, non-aqueous liquid phase, air, affinity to organic material. So many things can impact their environmental fate. That's why we have investigated how they will impact or how their fate will impact the soil ecosystem. Their distribution in soil depends to the many factors, including depths to the soil, hydrophilicity or hydrophobicity. The hydrophobic PFAS usually they have long chain. They are expecting to stick in the subsurface area, in the shallow area of the soil, and they will impact the soil, plants and microbial community, especially plants that they have a short root length. And of course, type of the soil, vicinity to source of contamination, all of them, they impact their distributions. The long chain, as I said, it will stick in the shallow root, between 0 to 30 centimeter. So these are long chain, that they are hydrophobic. But hydrophilic, they are short chain PFAS, mainly less than 4 carbons, that they go deeper, and when they may reach to the groundwater. Their toxicity of short chain is higher than long chain PFAS. Also the soil compositions. If they have more organic material or more clay content, their affinity to attach to the soil compartment is higher compared to others. And also they have shown the negative relationship to the pH. If it's pH higher, so they have less affinity to combine or attach to the soil particles. So they can differently impact soil microbial ecosystem. So studies, they have shown that they can cause oxidative damage, they can interfere with the membrane, transportations, and also it can impact the important enzyme that's involved in the soil, particularly some microbial community, they do nitrification in the soil, that their life would be impacted. That's why we have focused this research on investigating the impact of PFOA as one of the mother compounds of PFAS on microbial community in 2 different soil regimes. Flooded soil that simulates the wetland plants, including rice that's grown in rice paddy, and also aerobic. So the flooded soil, we are expecting to have 15% of oxygen, and aerobic soil 21% of oxygen, based on our result that we have obtained. And so this will help us to have better strategy for remediation. Microbial, we did two different tests. One, microbial viability, taking sample and counting the viability of the cells, counting the colony forming units on a Petri dish. Another techniques we have used, it was 16S RNA, to see how it will impact the diversity of microbial community, or shifting from different families of microbial community in the soil. This is one of our results that my student has done in the lab. This is a viability test. We are seeing that low concentrations, the impact is less. As we go to a higher concentration, impact will be extended. So we are expecting at high concentrations of the PFAS, we are having less microbial community. However, we have done another DNA analysis, 16S RNA, that brought better vision for us how the dominance of different microbial community will be impacted in different soil regime. You can see how the diversity will change, and how the abundance of different phylum and order of the microbial community in the soil will be different between aerobic soil and anaerobic soil, and each one will behave differently in exposure to PFAS. This is a heat map analysis. Again, they will show the shift of microbial